Let's get started. So I'm joined today by Jack Hittery and Nadia Heron to discuss accelerating drug development through AI. Jack is a serial entrepreneur and co-founder of several tech companies, including Sandbox AQ, where he's currently CEO. He's the author of Quantum Computing, an applied approach, and has been recognized for his leadership by organizations such as the World Economic Forum. Jack, can you tell us a little bit more about Sandbox AQ? Why did you found this company? What are you doing, and how did you get there?
Well, the vision for founding this company really goes back to my days at NIH, the National Institutes of Health. And I was doing research there, and our lab was one of the first to bring in the idea of neural networks, which is inspired by the brain's architecture, but we can instantiate them in a computer and use them for pattern recognition. We, in our lab, were using it to look at brain images themselves. So kind of a full circle, a brain-inspired pattern recognition tool looking at brain images. And what we showed is that indeed, we can have the computer see things that our human eyes could not see. So I knew from that point that this was going to be a critical tool to transform medicine and then many other sectors. I had to wait a number of years until the chips got better, until the software got better, data sets, and so on and so forth. But my vision of starting a company, a platform that would have as its core quantitative AI that focused on novel treatments, novel diagnostics, and then other sectors after that was born at that time. And now we have Sandbox AQ. The A, of course, Shoshana, is for AI, the Q for quantum, two of the twin engines that we use as superpowers to really solve very, very difficult problems. And I'm very proud of Nadia and the entire team for overcoming challenge after challenge and making a big difference already in quite a few indications.
Thanks so much. And that's a good introduction to Nadia. So Nadia Heron is the general manager of AI simulation at Sandbox AQ. She spent the last 15 years developing products in the life sciences and technologies fields for small and large companies, such as Johnson & Johnson and Google, which you may have heard of. Nadia, tell us a little bit more about your background and what are you doing now at Sandbox? How did you get there?
Yeah, sure. So when I first met Jack, he was actually at Google and he was applying his 10x thinking, which is what Google is known best for at the Moonshot Factory. And he was doing so across all these different domains that are really affecting many areas of our economy. And I was very attracted to him. And that was what drew me into Sandbox when I initially spun out. And now I lead two divisions focused on material science and drug discovery. And we are applying that same kind of 10x thinking. But initially, my career got started as a bench scientist. I started at Johnson & Johnson 20 years ago, setting up their proteomics labs. And at the time, that was the state of the art. And then I moved into what was a 10+ year career during medical devices all over the world and did that kind of work for Google. I specialize in the radiology domain. Once the radiologists were not as good as the algorithms, I went over to Google and did that kind of work. So really cut my teeth in the algorithm space at the cutting edge of where it was beating out what scientists could really perform for us. And that is very much as the ethos of what we do.
So maybe just to level set our conversation a little bit before we get into too much detail, I want to talk about AI and what that means for drug development. So for our audience, when a lot of people think about AI, they think about things like ChatGPT and other large language models. But when we use AI for drug development, we're actually learning on real world data sets, not large language models. So Jack, maybe could you tell us a little bit about the difference between using AI for daily activities or in the digital world as compared to AI for drug development?
This is a great question. I'll start off, and I'd love for Nadia also to add in information about, say, our data set, other data sets that we produce. But you're exactly correct, Reshaping. If you want to really impact the real world, right? If you want to say, hey, I'm not here to create another document, an image, a video. I actually want to develop a new molecule that's going to interface with some target in the body. And so let's say we're going after a certain cancer. We might be going after Alzheimer's, Parkinson's. We might be in the cardiac space. These are all very important indications. And traditionally, it's been really hit and miss, mostly miss, when it comes to traditional drug discovery and development. In fact, the numbers, I think, as many of our viewers know, are quite sobering. It takes generally 10 plus years to develop a drug candidate, and when it gets to clinical trial, 85% failure in clinical trial, and almost more sobering, if that's not already bracing, 40% of that failure is in phase 3. The most expensive phase, Shoshana, as you know well from your deep background in biotech as well. So when we looked at this issue of how do we really come to the table with new tools, we can't use a model that's trained on words, on social media posts, on cat pictures. That is not going to be the right tool for the job when it comes to something like as serious as a cancer-fighting drug or a diagnostic. And now what we need to do is have new data sets that did not exist before. This is not data you can download from the internet. This is not data you can just buy off the shelf. You've got to do real-world experiments and take that data and then supplement that with new equation-based data. People sometimes call that synthetic data, but I like to call it equation-based data to really make it clear where that data comes from. It comes from the laws of physics and chemistry. And we know those laws and we can augment these data sets. These data sets in turn can create new AI models, not the kind of chat GPTs that you're used to of typing a prompt in. Those are not really fit for purpose here, but novel architectures that are very well suited. Knowledge graphs, neural network models, and others that are really well suited. But I want to hand it off to Nadia to talk about specifically the kind of data we've been able to produce.
Yeah, I want to touch a little bit on the novel architecture. So if we think about the evolution of the GPU, right? So we borrow from our friends in video games. Those are graphics, right? This is the birth of the GPUs. When we think about the LLMs, that's a transformer model that predicts the next one, right? This is a 2D sort of like linear model. Now, when we get into images, right, generative images, now you get into like multi-matrix multiplication models, right? So we're looking at generating from a two-dimensional image, a little bit more complicated, so the GPUs evolved. Now the GPUs have evolved even more so where we're modifying the firmware of the thing that controls the GPUs to handle what's called triangular calculations, where we're looking at 3 objects in space across a matrix. That is highly complicated. This is the type of thing that people thought we had to wait for quantum computers. But it turns out we don't, right? And so through our partnership with NVIDIA, we've been able to exploit this kind of architecture through our models. And that is the power of the LQM. You cannot compare what is a two-dimensional fragment of letters to the complexity of a molecule that binds to its target in three-dimensional space, taking into account everything that's happening around it. And so that is what we've done. And over the last couple of months, we've released to the scientific community numerous models, including SARE, which has the largest binding affinity set of co-folded structures. There's a molecule, a drug-like molecule with its target that a person can use to start drug discovery, right? And so over time, we've relaxed the need to have the structures that you would have to go to a cryo-EM to get as an input to start drug discovery using computers. We've relaxed the need for that.
And just to build on what you said, AQ Affinity, which Nadia and team just released in the last 10 days to the world, public, open source, Shoshana, so that researchers can use it, drug developers can use it. AQ Affinity represents the frontier, the edge now of what is possible, both in terms of determining the structure of that new novel candidate molecule and the receptor, could be a protein as an example, and also the affinity, the binding affinity of how it will actually attach. So this is a very exciting moment. Again, large quantitative models, LQMs, versus and complementary to large language models.
Yeah. Okay, so that's a good transition. Most of us, I think all 3 of us, did get our start in the wet lab on the bench, and you typically would start exactly where you just outlined. I want to target a protein. It's an enzyme, it's a receptor, whatever it is. Let me look at the structure. Let me use medicinal chemistry to target a molecule. You know, oftentimes you have large libraries. It goes through the sequence, and the numbers are very disheartening. I mean, you threw them out there, Jack, but we're all used to hearing this, right? Approximately 10 years to develop a drug. I mean, once you know the target, even at that point. and less than 10% success rate. And I know I've been guilty of becoming complacent with that. You try to shave a year off, 2 years off, but I think we're at a point with technology where we can do much better. So can you help us to understand what are we doing with AI right now that can shorten the drug development timeline? Like, what should people be expecting today? And then I'll ask you about 10 years.
Sure. Let me start, and I'm sure has other examples, but when you look at the end-to-end of drug development, drug discovery, and then development, and also in the clinic, it's absolutely critical now to think about AI all throughout the process. Target identification validation, as you mentioned, can take years on its own, and that's an area, and I'll ask Nadia to delve into that. Once you have the target, you want to drug it either with a small molecule, or increasingly, a lot of us are interested in biologics, these larger molecules. We're having a lot of success there. And Keytruda and other checkpoint inhibitors are an example of a biologic that has literally saved the lives of millions of people out there. And then in clinic itself, people sometimes think, AI, its job is just to deliver it to the clinic and then we're done. Actually, what we're seeing is when something is in clinical trial, we find that of the failure, half is due to lack of efficacy and half is due to severe side effects. We call them SAEs, as you know, all too well, Shoshana, from your backgrounds as well, severe adverse effects. And often those drugs then go on the shelf, they get killed off, but actually we can now save some of those drugs. We can analyze what is that adverse effect or that SAE, and we can understand what part of the cohort of people in that trial are being impacted by that interaction. It's not everybody. Usually it's only a few percent. And often the trial has to be halted, and then we've got to analyze, and we've got tool sets now that help us understand what specifically is happening and then help the company make an assay that can test for that interaction before those folks come back into a cohort. We can then have that drug move on and have a very successful impact on the world without having to lose it in phase 2, phase 3. So Nadia, maybe you want to talk about the early stage as well.
Yeah, sure. So let me give an example of some of our work with Dr. Stanley Prusiner, who won the Nobel for the discovery of prion proteins. He won this Nobel in the early 90s, and he is now at a stage of his life where he doesn't have time to go through this process of potential failure. And so he came to us because he has this very novel mechanism of action, very novel modality. But as you know, when you're probing on the bench, you're looking at everything indirectly. You're looking at second, third order effects and trying to subsume what might be happening. And so he came to us and we built custom solutions to help him understand the complexity of his system using these physics-based methods. And I was actually in the room, I don't know if you remember this, Jack, when he first saw the mechanism of action of what was actually happening for what he had studied his entire career and what was going on in the brain. And he was like nothing short of shock to just fully understand that. And that's an example of kind of early stages, right? We've shaved years off that process.
It was an emotional moment, actually.
It was an emotional moment, yeah.
He got emotional.
He did. And then, for other companies that we're working with, they have drugs that are stuck in clinical trials. They need to show the FDA the mode of action because the FDA doesn't believe that what they're saying is true, right? They have no evidence. And so that's the kind of situation through simulation, we can shortcut those learnings. They can go give them the answer key, replicate that efficacy on the bench and show the FDA that data to move things along. So those are two very pointed examples. In the case of AQ Affinity that we just released last week, we can go from an amino acid sequence to a drug in complex with its target. That is novel. I mean, the Nobel was awarded 2 years ago for a static image, what is essentially a picture. It was not dynamic. And so it wasn't practical for the utilization across drug discovery for many people who actually used AlphaFold in drug discovery. Amazing innovation, amazing breakthrough, showed what was possible with compute, but we've taken it to its full depth of where it can go.
Yeah, that's really amazing. And, notes to self, we're getting ready at my current company to start our first clinical study, a phase 1 study
Excellent.
in oncology patients, and just recognizing all of the additional information that we can pull in through these technologies so that we can do our best job at predicting, the phase 2 that will show the best efficacy in the best patient populations at the right dose. These are tools we didn't have a few years ago.
Shoshana, if I can just add on that.
Please.
Another aspect of what these tools are impacting is who can now really make a drug. Traditionally, we've always thought of pharma and biotech. Those are the two typical places you make a drug. Maybe academia can help get it to a certain point, but then typically you'll be licensed out to a biotech or a pharma company. But now, with these new tool sets, it really democratizes and opens up the playing field. Yeah. We're seeing for the first time sovereigns, nation states, saying, hey, we've got a particular challenge, medical challenge in our community, in our population. We'd like to go ahead and get a drug developed. Now, they can take it to a certain part. For example, they can do phase 1 and phase 2 trials typically much faster and more efficiently than we can in the United States, because they've got state-of-the-art hospitals that they just built in the last few years. Yeah. For example, thinking about the GCC, the countries in the Gulf area, state-of-the-art hospitals, FDA recognized, really great docs there and clinical trial apparatus and setup. And they can then move it through phase 2 and then reach out to the pharma community and say, who wants to be our partner to take it to the next step? That is not something we've seen 4 or 5 years ago. Yeah. But these new tools allow non-traditional entities to be part of this incredible opportunity of developing key life-saving drugs, particularly when those drugs will impact diseases of their population that maybe some of the traditionals have not yet impacted.
Yeah, and what's interesting about that is that they don't have the baggage of the 10 years of investment in cryo-EM and state-of-the-art glassware and all of that stuff, right? They get to rethink this whole problem from an efficiency and an ROI perspective to help their people. And so in these cases, we're helping 2 particular sovereigns right now. And
I'm going to talk about Bahrain as an example.
Bahrain. So we actually just signed a few months ago a massive deal with Bahrain, where we're taking a drug end to end, and we're looking at something that we can make for them that affects the global population, but more importantly for them, something that affects their people endemically. Yeah. And that is just something that the economics of a rare disease is such that when you don't have the overhead to pay for, you can pursue it in a different way, and that's what they're exploring with us.
Yeah, I mean, sovereign nations are one thing. I'm thinking there's some academic physicians in our audience
Absolutely.
that might want to do that on their own as well, and it really opens up.
I think it does empower academic institutions now, individual labs, to take drug discovery much farther than they ever could. Typically, they'd have to stop at a very early phase, license it out, and really lose a lot of the control over the fate of that drug. Now it's really possible to take it to the next step.
Yeah. Okay, so what should we expect in the future? This is amazing what we can actually do today.
Yeah.
Where do you see things going 5 and 10 years from now?
Well, I can start. I mean, first off, we made a bet when we started this operation inside of Alphabet initially that the GPUs, the TPUs, as you know, Alphabet makes its own accelerated chip called the TPU. NVIDIA has the GPUs. We have partnered with both companies, both incredible operations and wonderful partners. We made a bet that it would go exponential, that the capabilities, the computation capabilities of these chips would grow and grow literally not even every 18 months, which is initially Moore's Law, but every 6-7 months. Yeah. And if you look at NVIDIA's roadmap from Hopper to Blackwell to Vera Rubin, and Vera Rubin, of course, an incredible scientist that they named it after, and that's also a wonderful thing. It was supposed to take 2, 3, 4 years for that evolution to happen. They've compressed it down to under 2 years in terms of that transition. So our thesis was correct back in 2017, 2018, that this capability of the chips would grow very significantly. We are also looking forward to the point where our partners who are making quantum hardware will now start to deliver to us scaled error-corrected quantum computers. That's probably not going to happen until the year 2030, 2031, but we're seeing really positive milestones being met by Google, by IBM, by Microsoft, by so many other companies, Quantinuum, IonQ. There's about 36 companies out there that have serious quantum computing efforts, and we test our software on a variety of those platforms. I think that will put more gas on the fire around the year 2030, 2031. But as Nadia pointed out, we're not waiting till then. Already today's chips with our algos, we're a software company, are able to really synergize and bring big benefit today.
Yeah, I would say this idea that we are going to wait till phase 1 or phase 2, 6 years after millions of dollars worth of investment to see a drug fail, that will be a thing of the past in 5
Great.
to 10 years. I
We'll
really
all be
do.
happy about
I
that.
think, yes, the IND enabling studies should all be done in silico. There's no reason why we can't do that. I will say Sandbox will do that this year.
Yeah, and already, Shoshana, if you look at, we had input into the FDA, many others did as well. This was a collective community effort about animal testing.
Yes.
And the main focus that we had in our conversations with the FDA and others had is that we're losing time and the animal models are actually not reflective of human biology. How many mice have we saved from Alzheimer's? How many mice have we cured diabetes? How many mice have we killed off a cancer? Literally thousands, maybe hundreds of thousands of mice. But the ability to transfer over to human biology is a wholly different story. And that's why people keep getting surprised in the clinical trial. Well, we tested in this animal model. Well, guess what? The in silico models are now better in many cases in terms of its fidelity to human biology than an animal model. And the FDA recognized this, to their credit, a number of months ago in a new advisory they gave out, saying that we, the community, could start to shorten and limit the amount of animal testing, go right from in silico, a little bit of animal testing, mainly for safety purposes, and then jump into the human.
Yeah, you can actually register your in silico tools with the FDA for other companies to use as you show their efficacy, and that is something that we're absolutely planning to do.
Yeah, so it's taken a while, Shoshana, but the change is starting to happen.
Yes.
Yeah, well, we'll all be thankful for that. I mean, within the drug development community, but I think the people that it will impact most are the patients at the end of the day, and I think everyone's waiting for that. But to your point, there's so much that we're actually able to do right now that it's amazing how far things have advanced already. So,
Absolutely.
all right, well, thank you both for
Thank you
joining
very
us today.
much.
This is